Well, hello. We're going to explore vaccines and biotechnology-based diagnostics and therapeutics. Um, I want to put a good amount into the vaccines, but I did put a bunch of links that I strongly encourage you to take time to view the lecture videos because I think they'll give you a much better approach and understanding of the complexity of vaccines as well as some of the fraudulent and quasi-science that is out there. So, without further ado, I think it's best if we just start up looking at what vaccines are. Um, when you look at the vaccine history, it includes triumphs as well as controversies. The first real sense of vaccines was varialization, and this was a Chinese technique, and it was an attempt to try to provide some sense of smallpox protection. What they would do is they would take dried and ground up smallpox scabs, and they would take these scabs and blow them up into the nose. This supposedly provided uh, immune defense against smallpox. As a matter of fact, when we look at the history of even the Revolutionary War, smallpox was something that was used. Smallpox was used as a bioweapon. And this goes back to Lord Amherst and some of his associates. What they tried to do was, because Native Americans were much more susceptible to the smallpox, they introduced some smallpox contaminated um, handkerchiefs, etc., and some blankets. And within a short time, the Indian tribes that were attacking a particular fortress were decimated by an outbreak of smallpox. Smallpox moved upwards. Now you have to understand that smallpox is serious. Um, in even the 20th century, smallpox fatalities was about 40% of those who were infected. Now, we jump ahead in time to Jenner. And Edward Jenner came up with a concept. He made these observations. There's a similar disease called cowpox. In milkmaids, yes, these were actually young women that would go out and milk the cows. They noticed that their skin was fairly pristine compared to a lot of the other people in the villages and towns, etc. Because usually what happens is that with smallpox, you end up with some terrible scars as a result of the poxing that occurs. So he surmised or hypothesized that cowpox must provide some immune protection to the milkmaids, and thereby they didn't get smallpox. So, as you can see in a figure in a minute, Jenner took some of the cowpox pus from a pustule and vaccinated a child with it. And it ended up that this little boy never got smallpox. Now, vaccination fears and the reemergence of old health threats are some of the problems that pop up with vaccinations. Now, let me help you to understand this. It's either cultural, religious, philosophical, techno ignorance. There were those that would say that certain vaccines caused autism, um, attention deficit disorders, and other types of abnormalities, and these were all explored. The problem that we have is that people don't always understand the vaccine technologies. The cultures don't always understand this. And also distribution of vaccines in war-torn and unstable areas of societies makes it very difficult. For example, The more recent situations with Yemen, they would have outbreaks of, I believe it was polio, and I think there may have been some measles as well. But because Yemen is under a civil war, it's very hard to get an established group of doctors with the equipment necessary to maintain stable vaccine cultures to vaccinate the entire population. And this is not just an isolated incident. Um, the other problem that you have is that some of the vaccines had to be in what is kind of called a cold chain. From the time that they're made, they have to be kept almost refrigerated until they're injected into the individual. This gets to be very difficult in developing countries where there is A, no electric power, B, no refrigeration, C, temperatures up to 140 degrees a day, makes it real rough. Okay? Now, here's an image, there's a picture done of the time. The woman that's bandaging her hand is the milkmaid who supplied the pus from a cowpox pustule, and they then did a scratch and an insertion of that pustule material into the little boy. And that was a successful time. This is an example of a child with smallpox. Now, if you notice, it's all over the face, etc. There are some cases of some types or strains of smallpox that the pustules would fuse and they would cover the face and they would go into the respiratory area. No, I'm not making this up. There are actual, if you look and do research, and there's a variety of books out there about the success. Um, one of them was entitled Scourge, and there's a variety of others, including the chronology of how they were able to eradicate this disease. There are only several repositories in the world right now that have any of the smallpox virus. And there's been a debate whether they should be destroyed. The problem is, do you trust them? And also, on top of that, there have been scientists in the first decade of the 21st century that were able to do work where they were able to piece together the genome for smallpox. The genome for smallpox is a DNA virus. They were able to stitch it together with just fragments here, fragments there. They would order certain special sequence links. And so the question there becomes, well, if we have the rest of the world that is not vaccinated, in the US, for example, they stopped vaccinating in, I believe it was 1972 or 73. So that means that anyone born after that point is not vaccinated. Now, in some cases, those who went into the military got vaccinated because it would be a potential biological warfare threat for smallpox. But they also get vaccinated against anthrax and a variety of other possible potential biowarfare weapons. But if you can imagine, as we're in this age of COVID-19, the susceptibility of a very contagious disease that leads to either scarring and or death um, to mostly a population that's not vaccinated would be a devastating event. Vaccines provided the means for people to basically get the learned immunity that was necessary to last them, and in some cases, last them their entire life. Now, if you take a look at this entire list, and the print is small, I admit, but what you're looking at is many of the successes of the development of vaccines. Some of them have not lasted quite a while. Others, you're probably looking at going, they got that? Japanese encephalitis virus vaccine? Do I need it? No. Because you see, you'd only need it if you were in an area where that disease is endemic, where it's native. Japanese encephalitis virus, if you went to, yes, Japan, and if you were there for a sustained period, yes, you might need it. At the same time, would you need cholera vaccine? No, unless you were going to an area where cholera was prevalent or where there was a threat of that. And this is some of the situations that arise where do you need it? No, or yes, why, etc. Now, most adults don't even realize they need to get reboosted once in a while. And I bring this up just to not get you scared, but to get you aware of this. My personal observation, a little sidebar here. In 2003, I was teaching microbiology. And I started going over the epidemiology section, and I started to come to this understanding of something. Wow, older adults, when you're talking 30s, 40s, 50s, people in the construction industry, for example, got a lot more cases of tetanus. Now, why would that be? Because tetanus does not last you for the rest of your life. You need periodic reboosters, roughly about every 10 years or so. Unfortunately, these individuals didn't get them, and in their later life thought they were immune, but got sick with tetanus and in many cases died. Therefore, like a lot of other diseases, you do need a booster from time to time. Not a lot, and it depends about the vaccine itself. Case in point, when the hepatitis B vaccine, which came out, it was a subunit, meaning it was a protein for the outer part of the hepatitis B vaccine, first was distributed to medical care workers. That was approximately 1985. And this was a really big move because Hep B was out there, and anybody that worked with blood or surgical materials or needles or anything, they just in general, surgery staff, there was a threat of getting Hep B, which meant either death or liver cancer or something else. In the beginning, they said you need a booster, you'll need it once or twice, and then after that, you'll need it every 5 years. Well, after 5 years, they came to say, well, okay. But 5 years after that, so now we're talking about 1995, you'd go up to the doctor and say, I think I need a booster for Hep B, and he'd say, well, it depends. Let's see. I'm going to do a blood test for you. Because if your titers for antibodies against Hep B are still there, you don't need the booster. Fast forward to 2005, when I'd be having a chat with my physician, he'd say, nope, you're good for life now. When the vaccines first come out, sometimes they're not sure, and they take the more conservative view, the more cautious view, to make sure that the patient is properly covered. As they have more data and more clinical information about the efficacy, the lifespan of these vaccinations, it turns out that they may not necessarily need them as often. I hope that gives you some thoughts. Now, by the way, there are other situations where you would need a vaccine, even though you think, can't happen to me. If you look, you will see on this chart the one for chickenpox. Okay? Now, that's the varicella-zoster virus vaccine, and kids are given it now. Protects against the chickenpox virus. But if you look below that, the herpes zoster vaccine, this is given for individuals, for people over age of 50. Now, some say 60. Now, here's the deal. If you had chickenpox when you were a young kid, well before they started to actually have a chickenpox vaccine, then you've got the chickenpox virus potentially sitting in some nice little part of your nervous system, preferably one of the ganglia in the cranial nerves. Oops. But if your immune system goes weaker and weaker as you get older, you have the chance that shingles will come back. In other words, the virus will come out, be reactivated, and it will come out as shingles, causing some very painful, uncomfortable situations. So now they have the vaccine, but it's oriented to older people. One other little problem, at least more recently, there's been a shortage of the vaccines, and you can't get it just once. You have to get once, and then about a month later, a booster. So that creates a bit of a paperwork and storage and handling dilemma for local physicians. Some places will have them, they'll offer them X, Y, and Z, but then you got to come back for that 30-day one to get proper immune coverage. So let's talk a little bit more about vaccines and also immunity that's acquired in a number of ways. First off, we talk about herd immunity and immunization programs. Now, herd immunity is really the community immunity, and that's based on the minimum number of the population vaccinated to prevent the spread of the pathogen to unvaccinated members of the population. Some people will just give a general bird's eye definition or, quickie definition of 85%. In some parts of the world, in some countries, in some diseases, 72%. Others will say 90%. Because what that means is, let's just say 90%, just to make it easy for numbers. 9 out of 10 people are immune to it. So if one person gets the disease, it's going to be numerically much more difficult for that disease to spread, to find somebody that is not immune, and then to cause them to be sick as well. Herd immunity gets to be a problem when suddenly a generation or a certain group refuses the vaccine. Then you have a larger cluster of individuals not immunized that could then be susceptible to the disease and thereby spread it to others. Now, the other thing you have to understand is, this is a side note, but one of the doctors that was working in the battle to eradicate smallpox came up with a problem. He was in Africa. There was smallpox outbreak, and he was trying to do at the time mass vaccinations, but he found he was getting short on vaccines. So he developed a new technique called ring immunity or ring vaccinations. Basically, what they would do is find the person sick, find who they were in contact with recently, vaccinate all of the people around that particular sick person, and they would stop the passage of the disease. In this day of COVID-19, that's partly how they're attempting, without a vaccine, to try to contain the outbreak by basically finding out who are your contacts. This is sort of an epidemiological approach. Eventually, if they have a vaccine, they would take on that as a means also. Here's one problem. Smallpox is different from some of the other pathogens because smallpox, and this is really the reason why it's been able to be almost eradicated, is there are no other reservoirs. It doesn't live in soil. It doesn't live in water. It doesn't live in birds or bats or pigs or dogs. Nothing, just humans. That's the only virus that's really been found. Okay, I don't get it from monkeys. I don't get it from this. I don't get it from that. I get it only from person-to-person contact, a person who has the smallpox. Okay, so if you have removed all possible persons who might be susceptible to the disease kind of like burns itself out. There's no other places that it would be able to be cultivated. Okay? Now, I'm also going to cover in a minute many of the different vaccine types. All of them have different pros and cons, but you have to be understanding of that. And it depends also when you talk about the microbiology of the organism. Is it a virus? Is it a parasite, such as a helminth or a protozoan? Or is it a virus? Is it an organism that spends a lot of its time intracellularly, such as a rickettsia or a virus? Or is it something that passes throughout the systemic circulation, therefore it would be much more susceptible to both a humoral immunity, such as B cells and the antibodies, as well as a cellular immunity, such as T cells, etc. Live attenuated vaccines, this is where you have a weakened pathogen organism, and it can still be used as a vaccine to stimulate the immune response, but by itself, because it's weakened, it can't cause the disease. Now, when we talk about inactivated viruses, inactivated vaccines, this is where you have a whole agent or a subunit, okay? A whole agent is sort of you could say it's a dead virus, a dead bacteria, etc. A subunit will consist of just some antigens that will stimulate the immune system, and the antigens were part of that killed pathogen, etc. However you look at whole agent versus subunit, these are used to stimulate the immune response. But here's a caveat. They may require multiple exposures, in other words, booster shots to fully stimulate the immune response and immune memory. Such as in the case I mentioned to you earlier about hepatitis B. I had to have the first shot, then 30 days later, the second shot, and then 90 days later, from my initial vaccination, the third shot. And the reason being was that I got 96% seroconversion, in other words, full immune response with the second shot. With the third shot, it was absolute. Everybody 100% had full immunity. So they don't want to miss anybody and then have them walk down the road and suddenly get the disease saying, but I got vaccinated. Now, there's a component that you have to also understand, and it's called adjuvants. Now, adjuvants are pharmacological additives. They enhance the body's immune response to the antigen. Usually, they're mixed in a small amount to that antigen. There are only two that are really allowed in the United States. One is an aluminum salt, and another is called phosphoryl lipid A. Now, the other thing that we have are purified subunit vaccines. These you have what is called the natural type and then the recombinant. The natural type, this is a purified subunit. It'll cause an immune response. But then we have the recombinant type. This is expressed via recombinant DNA technology. So basically put into a nutshell, let's say we take a virus. We want a surface protein on that virus. Now, some cases, a purified subunit is easy. You basically destroy the virus and you spin off and extract that natural antigen. But let's say you can't cultivate the virus, which was the problem originally in the 1980s with the hepatitis B. What they did was they extracted the gene code for the external protein, that is external protein on the surface of the virus. They put it into bacteria, and the bacteria just manufactured lots of this single subunit of that virus. It was not infectious, and then they purified it, and it worked wonderfully. Okay, so that's a subunit vaccine. Now, there's another one called toxoid vaccines. These are vaccines directed to the toxins made by the bacteria. For example, tetanus makes a toxin, diphtheria makes a toxin. So a lot of times what happens is when you're getting a tetanus shot, you're actually getting the tetanus toxoid. And this will be, of course, not at a dosage and not at a amount that would cause any toxic effect to the recipient. When we say toxoid versus toxin, a lot of times what it is that they have denatured the protein, if it's a proteinaceous, or they've done something so that it really doesn't cause any hazards to the recipient of the vaccine, but it causes an immune response. Now, conjugate or polysaccharide vaccines. Remember that bacteria will have sometimes, some bacteria have capsules, sort of a coating, makes it hard for the immune system to attack. Usually when they have these type of capsulated types of bacteria, they'll go after the polysaccharides that make up that capsule and make that part of a vaccine. Conjugate vaccine, these are vaccines linking a protein antigen to the polysaccharide, and this will help generate immune response. A lot of times these are more successful than just the plain conjugate vaccines. Now, there are new vaccines that are in development for persistent and emerging diseases. Two of them I thought I'd bring up. The There's a bunch of other variety of ones, and you can see this in your chapter. But the biggies right now are DNA vaccines. This is basically you take a plasmid, you put in some of the DNA for the pathogen, you attach it to the plasmid, and basically it's usually DNA for the pathogens, protein antigen, something, let's say, on the outside of the bacteria, the outside of the virus, whatever. You inject that into someone's muscle. The DNA gets into the muscle cells, and the DNA will then cause the host cells, muscle cells, to start secreting the required antigen. That will then trigger the immune response. Now, it's tricky because some people would say, well, wouldn't that cause the immune system to attack the muscle? No, not necessarily. Depends on how the plasmid is engineered, how the protein is secreted, etc. Recombinant vector vaccines. These are genetic materials of the pathogen antigen. They're inserted into a virus or a bacteria, which then triggers the immune response. Think of it this way. I'm going to take, let's say, well, since COVID-19 is out there, let me just take this one. I'm going to take the outside protein of the COVID-19, take the gene, put it into something as harmless as, let's say, E. coli, or a rhinovirus, a cold virus. And so the cold virus will now have that genetic information, express those antigens to the outside of the virus. You inject it into a person, and their immune system will respond not only to the cold virus, but it's like there's 160 of those. Being immune to one cold virus is nothing, but they will also have an immune response to that COVID-19 protein, and that should give them immune resistance to the virus. Okay. This is the concept of a chart that gives you the idea about several things. One of them is that if you notice here in green, there's no immunity here, this person's infected. Pretty soon the disease spreads and everybody's infected. Now, if we had this infected person, we've got some people here that are susceptible, and then we have several people here that are immunized. What that does is it reduces the number of people infected, and the people who are immunized don't get sick at all. This is the concept that we're moving toward with herd immunity. If most of the people are immunized, the disease usually can't spread, if at all, because mostly everybody's been immunized. Now, this chart, so you don't lose sleep on this, you're not expected to memorize it. I would like you to take a look at it to understand some of this. And for my wonderful nursing students, you'll get all of this in the scripts for vaccines, etc. But here's the main point. Here is the recommended pediatric immunization schedule that they recommend for hepatitis B, rotavirus, DTaP, which really is 3 different vaccines together: diphtheria, tetanus, and pertussis. Okay, and then look down lower and you'll see the MMR, measles, mumps, and rubella. When you get your first shot, and then you get a second one. Sometimes everything works out marvelous with just a single dose, like DTaP booster. Other times, you got to have more than one. Sometimes you have to have a third and even a fourth one, such as the pneumococcal one. All right? And here are how some of the various formulations occur. Again, you don't have to memorize things, but I would like you to be somewhat familiarized with them as we move forward. And here is the concept of where we make a genetically engineered one. And basically, as I said, you can see here's the gene for the antigen on the outside of this virus. We take the gene out, we put it into a recombinant vector virus, and this will basically create that same protein and secrete it out as you have cells infected by that particular virus. Remember that you have to have the virus here. The virus can't replicate, but it can infect some cells that are going to discharge the desired protein, which is your antigen, that the immune system will learn to consider as foreign and develop an immunity to. Now, the next area I'd like to cover for a little bit are immunological diagnostic testing. Now, here's the thing. Part of this goes back to some of the things that you've learned with innate adaptive immunity, as well as some of the immunological variances that you see. It's helpful to understand some of these from the techniques that have come from the basic research that you read in the prior chapters. For example, immunological diagnostic tests. They often rely on basically antigen-antibody interactions. Wow, yeah, I remember that. You may also remember seeing some of the parts about the serology, the study of the contents of the patient's serum, the plasma, agglutination reactions. Hey, that's something that you probably may have also remembered from your anatomy and physiology. Serology looks at just the serum, the plasma. Well, I have to separate that out from the formed elements, the red blood cells, white blood cells, and platelets. That's where I take my blood sample, put it in a centrifuge, which spins it around, and the heavier components separate to the bottom. The lighter components, which would include, of course, the plasma, stays at the top. You extract out the plasma and do further analysis, okay? For example, if you want to find out that there are antibodies present in that serum, well, antibodies will bind to antigens. For example, if you just take the ABO blood typing, you're not going to see an individual who's type A have anti-A antibodies, but they will have anti-B. And you want to prove that anti-A antibodies exist, you go to somebody with type B blood, spin out their serum, and you can use that anti-A antibodies to type A red blood cells. Okay. Now, immunoprecipitation. This is a test of antibody and antigen binding, which results in a precipitate in a gel matrix. Immunoprecipitation is a very effective strategy that we see with a lot of different types of tests, because what happens is that the antibody and the antigen come together and they will precipitate. They'll form sort of a really conjugated mess inside of this gel matrix. And you can see this in the radial immunodiffusion test, otherwise known as the Mancini method. Basically, you take a drop of your antigen and usually this is soluble, and some antibodies in a solution, okay? This is an immunoprecipitation test, and it quantifies the amount of antigen in the sample. The more concentrated the antigen in the sample, the larger the observed precipitin ring diameter, which I'll show you in a slide in a minute. Wherever you see this precipitin, in other words, this is where they're bound together, they're not going to go anywhere, they can't spread outward or diffuse through the gel any further, they're stuck there. Now, a double immunodiffusion test, known as the Ochterlony test, these are always interesting because you can test the patient's sample for various types of antibodies or antigens at one time. You put your sample in the center and you let other sample wells in the gel have the different things you're looking for, antibodies or antigens or whatever, and as they diffuse through that gel, and if they come to meet and bind, they stop there and you'll see this sort of cloudy area. There is another type of test called the neutralization reaction, otherwise called the plaque reduction neutralization test, PRNT. This is a test used to detect antibodies against a specific virus. Now think about this. If antibodies bind to the virus cannot infect a cell. What do viruses do? Well, I've shown you this even in live action. You will have a plate which has cultured cells on it. You put the virus into that plate, and what happens? The virus will infect cells. The cells will burst and release more new viruses, and you have what is called a plaque. Now, if you add antibodies that bind to that virus will not be able to infect the cells. And you will be able to use that antibody amount to determine how effective they're going to stop the virus from multiplying. Now, what will happen is the number of viral plaques will be reduced as a result. Here's the first one, this agglutination test. And you notice this is kind of like a micro test plate. And what you have is this. Keep in mind that you can take the antigen, attach it to these very small beads. And remember that because you have antibodies that have at least, well, this is IgG. Think about if it was an IgM, you have 10 binding sites instead of just 2. So literally you can have these Y-shaped antibodies, IgG. One will bind to this antigen on this bead, another will bind to this antigen on this bead, and so they all form this kind of clustered up agglutinated matrix. This agglutination here is a positive result. No agglutination here, that's kind of nothing. Ignore it, right? Okay. So you can determine and do lots of tests really quickly with various different concentrations, with various amounts, looking for various antibody-antigen interactions. Well, we did this before when we were in anatomy and physiology. You had type B blood, you had antibodies against type B, they bound up, they formed these agglutinations, these clumps. Remember the clumping? Now, if you had surface antigen B and you had antibodies against A, you wouldn't get any clumping. So there you get this big smeary pool of blood. Okay. Now, here's the Mancini test. Okay. You have the patient's sample, it's added in the well to the gel. You have a precipitin ring. This is where what's happening is the antibodies are binding to the antigen, okay? Increasing ring diameter. So as you have an increasing antigen concentration, the ring diameter continues. Here's the key point if I lose you. The gel contains the antibody. So you've got the antigen that you've added here, here, and here, okay? You can add different antigens, and which antigens will bind to the antibodies, okay? The more interaction you have, the greater the antibody interacts with that particular antigen versus this, versus this, versus this. Now you can do something even more cool. What you can do is you can have antigen B, antigen A. You can have the patient's serum containing antibodies. Now as they diffuse, okay, they diffuse through this gel and they come in contact with antigen A, they'll start forming this band here. And that's what you see right down here. This is the agglutination that they have between this antigen and this antibody. But let's say we also have another set of antibodies. Maybe this is from the patient's serum. So you want to check if they have a reaction to one particular disease or another particular disease. And what happens is that as the antibodies spread out here, they will interact with antigen B. This is why they call it a double diffusion test, okay? And where they meet up with and they precipitate out, they're going to stop migrating and they're going to form this cluster that you can see. It's kind of this band here of white. Now, this is where we got to that other one, the virus test, the plaque reduction. Without virus reduction, because there's no antibodies to bind to the virus, look at all the white spots. These are plaques. Remember that? So you take the patient's blood and you isolate the serum. And the virus is added to a series of serum dilutions. Okay? So you have the virus there. And here's the other thing. You're going to mix the virus with your antibodies. And a virum serum mixture, when the virus is coated with antibodies, cannot infect the cells. These did not have the antibodies there, and you can see all the plaques form. That's what those white areas are. The blue is basically where the cells are still remaining. But if you add the mixture of the antibodies from the serum with that virus, you'll notice that there's very few plaques. They're small and there's not as many. And that's the difference. The antibodies blocked the virus here from infecting the cells. Therefore, the plaques that exist are small, minimal, they're dispersed, but they're not as massive as you can see here. Now, the next one, again, this is a test that uses immune reactions, etc. This is called enzyme-linked immunosorbent assays, ELISAs. Everybody's heard of ELISAs. They probably can't tell what enzyme-linked immunosorbent assay, what the ELISA acronym means. This is what you're doing. You're setting up an interaction, and you're doing this so that you can have a rapid diagnosis. No more you're going to have to wait 14 days or a month or a few days. As a matter of fact, some of these interactions can tell you within a doctor's office. Basically, again, this technique relies on antibody-antigen interaction, but the enzyme-linked aspect depends on an enzyme that's going to act as a reporter. What I mean by that is some specific signal will say, yep, it's here. Let's face it, you and I can't just sit there and go, oh, look, it's an antibody. The thing is very, very small. But there's two things that you can do. You can make the antibodies very, very specific. That's where we get into the concept of monoclonal antibodies. These are identical antibodies. They will bind to a particular epitope, in other words, a particular part of that antigen, and only that antigen. And going back to 1975, when the researcher who developed monoclonal antibodies changed all sorts of diagnostic tests, because now what he had was not a bunch of different antibodies that could bind to this, that, and the other thing on the antigen, or this, that, and the other thing on the virus or the protein. He had one site. He had capability of making billions and billions of these antibodies. And that's what he did. Now, what's the difference, though, is you still got to have something that says, hey, it's here. And that's where we get what we call the reporter enzyme. Now, there's two types of enzymes. Usually it's horseradish peroxidase or alkaline phosphatase. Big, long words, very powerful enzymes. You attach them to the antibodies. And so what happens is one side of the antibody binds to the antigen, actually to the particular epitope of that antigen. The other part is attached to an enzyme. Now that enzyme is going to act on substrate, and the substrate either changes color or becomes chemiluminescent. In other words, it releases light. So in other words, it's either going to change the color on a microassay plate, or it's going to light up. And when that happens, you've got a positive response. Now, by the way, how do I know that it's always going to bind just the antigen? Well, what you do is you first add the antibody. You've already put in the plates the antigen. And then you give a little bit of time for the antibodies to bind to the antigen, and then you remove the excess, because the excess antibodies floating around would give you a false positive signal. And that's where I note right here. In each case, unbound antibodies, that's the first phase of them, have to be washed out. This is removing what they call noise, in other words, false signals. Now, there's several different types of ELISAs. Direct ELISA, indirect ELISA, sandwich ELISA. And then you got pizza ELISA. No, I'm sorry, it's a bad joke. You have sandwich ELISA. We'll explain all of them in a second. If you take a look at the diagram, which I'm going to have right here. Oh my, being difficult here. Okay, fine. Let me start off. The direct ELISA is the simplest format. It allows for the detection of antigens in the sample. Okay? Usually you put the antigens into the sample cup or well. You add your enzymes you add the antibodies that have labeled with the enzyme on it. And then, after you've washed out the unbound antibodies, you add a solution that will react with the reporter enzyme. If that solution reacts with the enzyme and it changes color, you got a positive because you have retained the antibodies because they bound to the antigen. Antigen's here, you're positive. Now, the indirect ELISA is a tool for determining antibodies to a particular antigen in a sample. In the indirect, there's a similarity, but here's what you're going to do. They sometimes call it the double antibody test. What you have is you have one set of antibodies. They will bind to the antigen. The antigen is already in the well or the little cup. Then you add it and you remove the unbound ones. Then you add a second number of antigens that are specific for the first antigen. Excuse me. You add a second number of antibodies that react to the antibodies that are bound to the antigen. I know it can get confusing. Trust me, it takes a second. This second antibody has attached to it an enzyme that will light up. Now, the advantage there is you can get a bunch of different second antibodies binding to the primary antibody, and that will increase the signal, okay? The sandwich ELISA, much more sensitive. It can require the two antibodies, and these can be done, the detection one, which with the enzyme attached. That's what I was talking about earlier also with that indirect approach. But the key point you have to keep in mind with all of these is that it can be done by a plate reader. Now, guess what? The pregnancy tests. They're looking for the antigen HCG, which is human chorionic gonadotropin. That shows up in the urine of women or in the blood of women within about a few weeks into the pregnancy. Initially, it's made by the tropoblastic cells on the exterior of the embryo. Then it's taken over later on in the pregnancy by the placenta. This replaced the rabbit test. For those of you that probably are far too young to remember, yes, they actually would take mom's urine and inject it into a rabbit. And if the rabbit ovulated, and of course that meant they had to kill a rabbit, they would look at the ovaries, and if the ovaries showed up, they would say, yes, you're pregnant, congratulations. Unfortunately, the rabbit didn't have much to say about that. But this test doesn't require a plate reader, and this is one of these very simple things you consider at the home pregnancy test. You basically discharge some of the urine on one end of the tab. The urine with the HCG will go up, and you will bind, and then it will have a second binding, and that's where you get that second line that indicates, yes, you're pregnant. Now, as we've gone through some of this, and I apologize if it was a little bit confusing, what you have is a variety of immunological techniques, usually by relationship of antibody and antigen. There is one called immunofluorescence microscopy. And this is a protocol where fluorescently tagged antibodies recognize a specific antigen. And this is where you incubate the antibodies on the cell or the tissue sample. Basically, it's almost as if you were saying, well, aren't you like staining the cells or the tissues? Yes, but in this way, I'm doing it with antibodies. And the antibodies will do what is called immunofluorescence. They'll basically light up using a special microscope. What the microscope does is provide a specific frequency of light, and the antibodies will have a fluorescence molecule, and it will give off light. And you will say, oh, okay, so the target that I'm looking for let's say it's a pathogen, a virus, which has a certain antigen, and the antibodies bind to that antigen. So wherever that you have lighting up, you have the presence of that target antigen and that target pathogen. And they gave an example of a rabies virus in a brain tissue sample. Okay, and this is very commonly used when you have situations, for example, if someone has been bitten by an animal, they will take the head of the animal, send it down for testing, and if they find the rabies virus in the brain, then the individual has to go through the rabies vaccinations. Now, the second one is interferon gamma release assay and IGRA, which is a mouthful. This is used to detect tuberculosis infections. And what you do is you're exposing the patient's T cells to TB antigens and measuring the amount of interferon gamma released. That's what INF gamma release means. Okay? Here we have the direct ELISA, and you can see where the antigens are in the test sample well. You add the enzyme link detection to the antibody, and this is where I hope I make this really clear now. So this is direct ELISA. Antibodies bind. Antibodies have an enzyme on the edge. Take away all the unbound stuff, add a signal that interacts with the enzyme, and that will give you the change in the color. You notice here on this plate reader, some of the wells are yellow, some of them are not. They call this the reporter enzyme, which is what you have right here. The reporter enzyme will convert that colorless molecule to a colorful molecule. Indirect ELISA. Again, you've got the antigen that's bound to the bottom of the wells. You have the patient's antibodies bind here. Now, this is different. First one, we're looking for the antigen. This one, we're going to be looking for the patient's antibodies. And then what we do is we have a second set of antibodies added. These have the reporter enzyme here. The reporter enzyme bound antibodies will bind to the patient's antibodies here. You clear off any unbound antibodies each step, boom, boom. And then you come in here, you add the signal chemical, it gets converted by the enzyme, and there you have a positive result. Now, with a sandwich ELISA, and I apologize, I was a little bit off there. Here's how we go. First off, this case, what you're using at the bottom of the well is a capture antibody. It is going to be targeted to capture a particular antigen. So we've got antibodies on the base. We add the target antigen. Okay. We then would clear out any excess that's not bound. But now what you have is antibodies, antigen. Now you're going to make the sandwich. And the sandwich is based on this premise. You've got antibodies that capture the antigen. You have the antigen. And now you've got the enzyme link detection antibody. Here's the enzyme part. Here is the rest of the antibody. It binds also to that antigen. So what we're looking for in this case is the target antigen that's retained by the capture antibodies and detected at the same time by the enzyme linked antibody. You add the signal, you'll get the color change. Now, here is the other test kit. And it's a pregnancy test kit. I mean, there's some students out there that might recognize it. It's like, wow, yeah, I remember that. Now, here's the idea. If you have that straight line here, that horizontal line, you're not pregnant. What they have is either urine, in this case, I believe it's urine, would drip on the stick. The urine and its associated molecules, including HCG, would travel up along the porous material here. And right here would be antibodies against HCG. So when they bind, they'll either cause a chemical or color change, and you will get the plus formed. Real simple there. This other image, which looks like a spattering from some modern cosmos picture, it's not. What you have here is a brain tissue section. Now, here's the deal. You don't have to process the tissue and extract this or that. You take a thin slice of the tissue, you put it on a slide, you add fluorescently tagged antibodies, you cultivate it or culture it, incubate it for a little bit of time to allow the antibodies to find the antigen. And if they do, you take the same slide and you'll see where they accumulate. Yes, you have to wash off the unbound antibodies, but these are going to fluoresce. The basic idea of fluoresce is that you have light at a certain frequency, it hits the molecule responds by giving off light at a different frequency, and so you have this color dots indicating the presence of, yep, rabies in the nerve tissue. Now, another area that we wanted to move on to relatively quickly is looking at selected genetics applications in medicine. The first one, and I have to admit, it's one of my favorite ones because I learned it quite a while ago, is the polymerase chain reaction. As a matter of fact, only about 6-8 months ago, the inventor of this, who got the Nobel Prize for it, Kary Mullis, passed away. PCR can be used to diagnose infections and genetic disorders. Now, here's the problem we had. If you think about it, we had it a little bit with COVID-19. Some people would be infected, and they wouldn't get symptoms, maybe up to 7 days, sometimes up to 14 days, okay? But they're infectious to others during that full period. So they haven't seroconverted yet to show off antibodies to the pathogen. So how do you define if somebody is really infected or not? Now, by the way, this is not an unusual phenomenon, okay? There are a variety of diseases, smallpox, and a few others, that basically you could have an incubation period of up to 21 days before you see symptoms. And by the time you see symptoms, you may have exposed other people to the same virus, bacteria, or whatever. HIV was also one that in its history was a real difficult one, because the first test that was based was an Eliza one, but they weren't looking as much quickly for the virus, they were looking for antibodies. But the problem is that up to 6 months, some individuals would not show antibodies to HIV. Oops, and yet they're infectious all that time. So this comes down to an issue I used to give to some of my other students in biotech. The haystack or the needle. You've heard of somebody saying, well, that's like looking for a needle in a haystack. Okay, fine. What if I had a better way to look for needles? Or I could make a lot of haystacks, each with a single needle in it, and maybe I'd be able to find that needle. And it ain't going to work. I need to make more needles. I need to make more of something that really will be the target and an identifier of the disease. Now, we're not making antibodies yet. It may be a while, so what do we have? We still have the infectious agent and its agent's genetic material. Its gene, its DNA or RNA. And that's where PCR comes into play. It's a DNA amplification technique. It creates billions of copies of a target gene in just a few hours. Now, there's a device called a thermal cycler, just so that you're familiar with it. It's basically a machine that provides heating cycles and cooling cycles. And in these periods of time, you will be able to take, let's just say it from DNA perspective. What's DNA? Well, it's a double-stranded nucleic acid. Okay, that means I'm going to have to melt apart the two strands so I can get access to the A's and C's and T's and G's. Yeah. And then I'm going to have to attach some primers that happen to lead on DNA polymerase to start making copies of the exact sequence of DNA that I want. Okay? That's where we get melt, anneal, amplify. Okay? Amplified is the last step where the DNA polymerase would be going chung, 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 chung. Okay, I've got one open up strand. I want to look for it. It's a cancer gene, or it's a virus gene, or a bacteria gene. It's unique, and I'll just make copies of this. Now, if I do these cycles, and you have like one cycle in a period of 90 seconds, in a period of 3 hours, you can make literally a lot of copies. How many? Okay, let's just say I can make, I think it was something to the effect that if I do the numbers and I have 90 seconds for a full cycle, 30 seconds for melt, 30 seconds anneal, 30 seconds amplify, basically in a few hours I have well over 1 billion copies. That's where I meant where I could get a lot more copies of my needle. The heck with it, I could have a huge mountain of needles. I'd be able to really identify those from the haystack, right? So the key thing I need is a very, very tough, resilient DNA polymerase. Now this one is called TAC. It really was something that became sort of the workhorse. And it was devised in a way with Kary Mullis looking at this saying, look, I need a DNA polymerase. It is a protein. When I heat this up to a certain temperature, under normal conditions, the normal DNA polymerase denatures and it's worthless. Then he found from an archaea life form, remember those that are they live in the extremophiles, they can live in like hot springs and volcanic, undersea volcanoes and all these other things. There was a particular bacteria called Thermus aquaticus bacteria. They were able to isolate the DNA polymerase from that, and it has a tremendous amount of thermal stability. And yet it's a great DNA polymerase protein. So what you do is you take your target DNA, you take free nucleotides, because you're going to build new strands of DNA. You take your DNA polymerase, you put it into your thermocycler. You have a melt cycle that lasts a short time, separates the DNA double strands. Then you cool it down somewhat and you anneal it. You're going to have the specific DNA template primers attached. These primers are necessary for the DNA polymerase to effectively attach on the strand where you want to copy that part of the strand. And then we do the amplification. And we go up to another level temperature where the DNA polymerase can just basically like Pac-Man walking along, walk, 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 walk. It's going to basically look, make a copy of the opposing strand. And you will be able to make strand after strand, because what happens is you amplify, then you melt, anneal, amplify again, melt, anneal, amplify again. You started with one strand of DNA, looking for a particular sequence of that, and you start going 1 to 2, 2 to 4, 4 to 8, 8 to 16, and I can go on and on and on, and I don't want to do that. Now, there's another type of PCR that's been more innovative. It's known as quantitative PCR, qPCR, otherwise referred to as real-time PCR. This method, you can quantify or measure the amount of copies of a particular gene present in a sample from the start of the reaction. So in other words, what it says to you is, that's nice. Let's say I only have one copy of the virus in a deciliter of blood. Well, one can eventually be many more copies of that virus. I need to know if the person is infected with the virus, period. So I'm going to have a really high sensitivity. Or I have so many cells here, which one really has that particular gene in it? There's a way to use that, and that's what real-time PCR does. It uses also fluorescent imaging to visualize the DNA copies as they're being made, which is really cool. Now, some of you might be saying, look, these are all small little things, and how do I separate them? You've probably seen the different bands of DNA on like a gel or something like that. That's because of the result of a technique called gel electrophoresis. It's a technique to separate DNA strands by size. Think of it this way. You have this material called polyacrylamide gel. It looks solid, but it's not. Think of it as a massive sponge with big and small holes, etc. And as you have an electric field push the DNA strands through the gel, the longer pieces are going to take longer to get through the gel. The smaller pieces move faster. And eventually, you set up bands, each containing sequences or sections of DNA of different sizes. The fastest ones are the smallest pieces. The slowest ones are the bigger pieces. And from that, you can literally cut that piece out of the gel, examine it, resequence it, identify it, put it into a cell even, and make the cell produce the gene product. It's really cool. Now, there's another area I wanted to bring up, and that was called reverse transcription PCR. Now, reverse transcription you might be familiar with. You may have remembered that going, wasn't that with, didn't we have something called reverse transcriptase? Oh yeah, we started off with RNA and we made a copy of it into DNA. What if you're going to do PCR, but you love to do PCR for RNA? Now, you can say, well, I have an RNA virus here, sure. But also RNA, the number of RNAs for a particular gene can tell you how active that cell is in making that particular gene product. A lot more RNAs, a lot more product being made. Very little RNAs made, very little product made. Okay? So let's say we're just looking for the genome of an RNA virus, COVID, for example. The PCR technique will allow you to do that. You take your sample, you add first reverse transcriptase, so you convert the RNA to DNA, and then you move forward with the PCR process, making up a lot of copies. And that way you're able to isolate the gene from the rest of the genome and do all sorts of neat scientific work. Here's PCR. Primers, nucleotides, Taq polymerase. Here's the area of interest. I've got to add a primer here and a primer here. This is when I had cycle 1. Part of it I have to separate the strands, so I have to melt them. Okay. Then I get the next cycle part, which is annealing. So I have my primers attached, and then amplification. Taq starts going down in a direction, this way, in a direction, this way. And then it stops. That's one cycle. Oh, wow. One cycle I have made went from 1 to 2. Cycle 2, I now go from 2 to 4. Cycle 3, I went from 4 to 8 to 16. So you can see how quickly it can work. And as I mentioned, a lot of the drug development that you see today is partly the result of recombinant DNA techniques. When I say recombinant, I'm putting a gene from some organism into some other organism. Usually, for example, bacteria, you take something complex and put it into bacteria, bacteria make a whole mess of it. For example, that hepatitis B subunit that I wanted to make. Now, you could do something else. For example, we have Humulin now, which is human insulin. You know, anyone that's a diabetic, everybody goes to Lilly, Eli Lilly, and gets their Humulin. Why? You see, what happened was before we had diabetes with insulin treatment, the diabetics died, sadly to say. But then what they started to do is say, look, we've got slaughterhouses all over the country, and we slaughter pigs and cows every day. We could use those pancreases that we've normally been throwing out and extract out the insulin. And they did. So for a while, going into the 50s and 60s and 70s, people were using either cow or pork insulin. Not bad, not perfect. Sometimes they would get a resistance, so they have to switch from cow to pig or whatever, but it kept them alive. And then in the 1980s, they started working on several different approaches to make human insulin. They had isolated the insulin gene from humans, and they now were going to put it into a plasmid, turned out not to be bacteria. They had to put it into a mammalian cell line. I think it was Chinese hamster ovary cells, because the bacteria would not provide for certain attachments on the sides of the two strands that make up insulin. But what you do is similar: gene isolation and copying, inserting desired gene into a plasmid, taking that plasma and transforming the cells into cells for expression. In this case, they would just use pulsed electric charges to open up the cells. Sorry, it's not changes, charges, to open up the cells for transfection. When the cells receive these plasmids, they would integrate them and they would start reading off the information, replicating the gene product, which would be an RNA, and then eventually making it into proteins. Okay? There's one other thing that the book brings up, which I didn't want to spend too much time on, but I want to just briefly. When you add sequences of DNA, you end up with problems because, remember, you've got to have base complementary base pairing, and that is where basically A's will connect with T's, C's will connect with G's. Now, if you have a strand of DNA and another strand of DNA, they've got to meet together and either be glued together at the sugar phosphate point, and that's if you've got blunt ends, or if they have an overlap on each other, you've got to make sure that both overlaps will agree. So if one part says CAG, the other one has to say GTC, if I got it right. And always you'll have to include also DNA ligase, which is kind of like the glue that kind of attaches the strands together. Here you can see what I was talking about earlier. We made up lots of copies of this sequence through using PCR. We then use certain enzymes that will cut at certain sequences of the DNA. We have the similar cut sites here in our plasmid. We attach the specific target DNA into this plasmid. We use the enzyme DNA ligase to bind them all together nice and tight. And now what we've got is what we call a recombinant vector. We put this into the bacteria makes a lot of it. And now what we've got, since we originally took this from a virus, we've made viral protein, which can now be made into a vaccine. Sorry about the duplication there. Anyways, finally, one thing that has taken a lot of attention today is CRISPR. CRISPR is an acronym standing for Clustered Regularly Interspaced Short Palindromic Repeats. Yes, I'm sure if you said that 5 times fast, you would stumble all over the place going, huh? What this is a means to edit genetic material. There are 2 components. But best known procedure is called the CRISPR-Cas9 system, which is Cas9 stands for CRISPR-associated protein 9 nuclease. Basically, this is a gene editing tool that has caused a lot of excitement. Yes, they can get excited in the medical world too. Because what you have now is a device that can act as a locator for the gene site to be removed. That's the CRISPR component. This enzyme cluster called Cas9 acts as the molecular scalpel. It will cut out the sequence once it's located. And guess what? You can also put in a corrected sequence with this technique too. And as you can see here, so there's a mutation responsible for a disease. Okay? So what you do is you use the CRISPR-Cas9 and it comes in and you have the cut points and it basically will remove, it'll identify first, then remove the part you want out, and it will add in the replacement. They're just starting to look at this in a variety of ways for human studies. So it's not like tomorrow morning they're going to have on the front page of the paper cures for A, B, C, D, E, F, G. Because like anything, you have to go through clinical trials, you have to go through clinical studies, you have to be able to understand the risks, if there are risks existing, and also how to deal with them. Now, in the last part, I want to just go over a couple of other small points. Some of this will require you to take a little bit of a review back to page 159 for review. We talked before about viruses that can deliver genes to human cells. We found that we know that viruses can carry genes into bacteria chromosomes. Now, some of the gene techniques, aside of CRISPR, have attempted to do this. Let the virus act not an infectious means, but to deliver a corrected gene, and the virus itself will integrate itself into the chromosome, but not cause any disease. Okay? That's the basics of it all. Has it been as successful? There's been a few cases of some success, but it's limited. And some of it depends on a lot of important ethical and medical review before such tests are done with human beings, because in fact, sometimes when some genetic material is inserted, the big fear is it could be inserted into the wrong area, the wrong gene, and shut off other genes and literally make the patient more sick, if not kill them. Now, there is a small section that talks about genomic mapping, and that reveals a lot of information. Since the end of the Human Genome Mapping Project, which started approximately 1987 and was declared completed basically in 1999, we have seen an explosion in information of not just the human genome, but a lot of other organisms, etc., as well as pathogens. Because as we were ramping up in our mapping strategies, we were developing newer, quicker, faster, more accurate reading technologies and techniques. So we could identify a lot more of the genes, not merely in humanity, but in a variety of pathogens. This is where we get into the concept of pathogenicity islands. It's not like an island of Gilligan's Island, trust me. It is a special grouping of genes. A lot of these are clustered around each other, and this is the problem. They provide virulence factors. They might neutralize the effectiveness of white blood cells. They might have a capsule to protect themselves. They might form a biofilm. They might release toxins that affect the patient. Usually, this strategy is acquired by horizontal gene transfer. In other words, bacteria coming up and trading information. A lot of times, this trading occurs via plasmids that have these gene factors. Sometimes the plasmids then get integrated into the full chromosome of the pathogen, like bacteria. Why is this so important? Because it helps us to understand how and why we're getting a variety of new factors that we're seeing with diseases that we've studied for 50 years, 100 years, etc. Also, for example, it gives us an idea about the pathogens that we're dealing with now and how they are now resistant to a variety of antibiotics. It's just something to think about. Finally, we just wrap up with gene microarray technology provides a global view of cellular functions. One of the things with microarrays is this. You can set up on the array information, and you can tell certain genes are expressed and their degree of expression from a patient's sample, because this gives us an indication of individual differences, how they react to diseases, how sick are they with this same disease, and what defenses they have against this disease, or how vulnerable they are to that disease. So the arrays are there. I'm just trying to bring this up as just a general coverage. Here is the situation I mentioned to you before of a vector virus carrying in a particular gene to help replace a defective copy of a gene in a human cell. This is just an example here. Now, the P stands for plasmid, okay? This is a simple plasmid. It's about, let's see, based on the sequence map and everything else here, I'm going to say it's roughly less than 5,000 base pairs long. But look at this. This is one thing that's been found. It has a gene for tetracycline antibiotic resistance. It has another gene that regulates how many copies of the plasmid will exist in a bacteria cell. Its origin of replication, this is so that it continues to be replicated when the cell undergoes binary fission. That's right here. But notice one other thing. Here's a gene for ampicillin antibiotic resistance, all in one nice, neat little package. If this gets transferred from one type of bacteria to the other, that is partly how we are having this crisis that's building up of antibiotic resistance to a whole host of diseases. It used to be you go to the doctor, he'd either give you the shot or he'd give you the prescription for the pill. You'd go home. So many days later, you feel better. Now, you curse the doctor thinking he was stupid and he gave you the wrong drug. Guess what? It's not necessarily the doc didn't know that. A lot of them are very savvy and they'll say, if this doesn't do anything in 5 days, call my office, we'll give you something else. Because he recognizes the potential that some of these pathogens now are resistant to various types of antibiotics. And if he's got to switch you from one type of antibiotic to another, that may be the effective strategy, but it depends also on patients giving feedback. We'll get into antibiotics next. Oh, yes, and here's this little bit here about tumor. They took a tumor biopsy and they did a microplate reading, and the areas that were fluorescing the greatest were telling you which genes were being expressed at the time, not putting a heavy amount into the microarrays. But where I wanted to come to end this is it's not an adaptive immunity. I'm sorry about that. This was a goof up. This should say vaccine. Okay. But anyways, I would like you to review the visual summary on page 430. I would like you to review the chapter overview in 431. Take time for the end of chapter questions on 432 and 433. Please don't forget your homework in Mastering Microbiology and your quiz in Mastering Microbiology. Peruse the videos in Blackboard for the lecture content. The next section we are going to do is Antimicrobial Drugs, chapter 15, and that will prove to be very timely in this time and age. I hope you have a nice day and a nice week.